Assalamualaikum and hi to everyone. For this lab is about fish hematology. So the title for this lab is the characteristic of fish blood, red blood cell in fish hematology. So let me introduce myself. I'm Nurul Suhaila binti Maksman. My matric number is S62929. So for the introduction, a fish blood consists of blood cells and plasma, similar to other vertebrate animals, and the blood cells are composed erythrocytes, leukocytes, and thrombocytes. Erythrocytes, that is red blood cells, are a round or ellipse-shaped cell and contains dense hemoglobins. Hemoglobins in the blood transport oxygen from the gills to the rest of the body, include example the tissue where is released that oxygen for cell use. I will pass to.
Hai, Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Haziq bin Muhammad Nurmalah. My matric number S61697. So my part is research objective. So for the first is student can learn how to take a sample of blood by using a syringe. Next, to understand the function of the circulatory system of the fish. And the last, to know about the handling process of fish when fish live use. Next part is research hypothesis. So the research hypothesis of this lab is different species of fish will affect the type and structure of the blood in the fish. So that's all my part. Thank you.
Hi, Assalamualaikum. My name is Nur Arisana Suha binti Mokhtar, S81405. So I will talk about my part, which is the result of this lab. Here are the results from the blood smear of tinfoil under microscope using 40 magnification and 100 magnification. Here are the results from the blood smear of tilapia under microscope using 40 magnification and 100 magnification.
Hi and Assalamualaikum. My name is Nur Atira Husna binti Ahmad. My matric number is S62801. Today, I will talk about what is blood and the function of blood smear. Blood is nutrients, hormones, minerals, immunological components, microbes, water, gases, poisons, and waste products are just a few of the various components that fish blood conveys. The blood smear's purpose is unknown. An examination of the blood, known as a blood smear, reveals detail on the quantity and composition of blood cells. In conjunction with or as part of a complete blood count, it frequently performed. Sickle cells anemia is an inherited condition in which the production of aberrant hemoglobin or the red pigment found inside of red blood cells occurs. That's all from me. Thank you.
Hi, my name is Imam Faris and explaining about comparison with two species, typhoid bud and tilapia. Okay, for the type of red blood cell is same between tilapia and typhoid bud, which is acroctocyte similar in form and ultra-structure to those to other non-mammalia vertebrate, teleost, fish and erythrocyte are a type of red blood cell. Normal teleost blood cell morphology matured erythrocyte. The shape of RCB is normal and matured erythrocyte. Because the shape of immature erythrocyte is typically more rounded than oval, with a blue tinted to the cytoplasm and a bigger, more heterochromatic nucleus, resulting in a greater nucleus to cytoplasm ratio. Teleost blood cell morphology, matured erythrocyte have nucleus, dark spot at the middle of cell. The nuclei of the cell are positioned centrally and are moderate to deeply basophilic. The cells are oval to elliptic in form with abundant pale eosinophilic cytoplasm. Normal teleost blood cell morphology, matured acrocyte, which acrocyte are ellipsoidal nucleus cell. Okay, for the net is nucleus cell have organelle in their cytoplasma, unlike mammal red blood cell. Fish red blood cell RCB have organelle in their cytoplasm. That's all from me.
Hi there, Assalamualaikum. My name is Muhammad Irfan Daniel bin Roslan and my matric number is S62834. Today I want to present about the discussion. For the discussion is the world's fastest growing industry for producing food is aquaculture. Today, 50% of the fish consumed worldwide come from aquaculture, and by 2030, it's expected to be the main source of fish. Due to the growth of aquaculture, the use and validity of fish health monitoring system have come to light more and more. The complete blood come to light more and more. The complete blood count CBC is a crucial and effective diagnostic tool and a part of a minimal database. It can be used to track change in fish diet, water quality, disease, and respond to treatment as well as change in their overall health. As a result, for Orichromys mozambicus and Barbonimus wenenfeldi that show the blood type. Looking at the picture, some blood cells are close together while others are far apart. This occurs because there is a chance that a mistake will be made through to this process. The procedure used must be accurate if you want ideal result. Besides that, because the automatic method used for mammals are typically not appropriate for fishes, The examination of blood parameter in fish has up until now been done manually using a Navier and Becker hemocytometer because of the presence of nucleic acid. The manual blood count has many potential causes of mistake which can result in wrong interpretation of the blood cell count. For example, improper mixing or dilution of blood and stain and improper charging of the hemocytometer chamber. Fish has been used to produce an automatic blood cell count, although very few research have written about this particular technique. That's all from me. Thank you.
Assalamualaikum, I am Putri Nakibah binti Abdullah S62918. I will talk about conclusion. So, in conclusion, unlike mammalian red blood cells, fish red blood cells are nucleate cells with organelles in their cytoplasm. In addition to their well-known function in gas exchange, nucleated red blood cells have recently been assigned a number of additional biological tasks connected to the immune response. Nucleated red blood cells can phagocytose and serve as antigen-presenting cells. Fish have cold blood, ectothermic. This means that their body temperature is significantly changed by their environment rather than being maintained at a steady level. Red blood cells have cytoplasm that is either light blue, coppery pink, or coppery purplish. On preparation stain with Giemsa, mature erythrocytes appear pink or yellowish and contain a lot of hemoglobin. So that's from me. Thank you.